Class II bacteriocins are membrane-active peptides produced by bacteria of all genera that have antimicrobial activity against several pathogens. They bind a specific receptor protein in the membrane of target bacteria, forming pores that produce permeabilization and cell death. However, it is not known if the pore is actually the receptor forced into an open conformation, or if the receptor just acts as a docking molecule, allowing the insertion of the bacteriocin to form the pore. Some of the bacteriocins we work with are Entrecin, which is produced by and is active against gram-positive bacteria, and it uses a MAM-PTS complex as a receptor. Microzin B is produced by and is active against gram-negative bacteria, and the receptor is a protein SEAC. To reveal which one is the best model, we built the suicide probes. We designed hybrid genes that encode a fusion between a membrane protein called ETPM and the bacteriocin. When these fusions are induced, the bacteriocin portion is placed in the outer face of the membrane and remains attached to it through ETPM. When the host cell lacks a specific receptor, the fusion kills that host. This means that the receptor is not essential for the pore formation, and that is why these fusions were called suicide probes. This system is expressed in E. coli because it naturally lacks the MAM-PTS receptor for Entrecin, and we also used a mutant that lacks the SEAC receptor for microzin B. We developed control system as well, one that only expresses the ETPM protein to make sure that the toxic effect is due to the bacteriocin portion and not just because of the overexpression of a membrane protein. And we also used the TAT system that exports the bacteriocins to the periplasm of E. coli to make sure that we are working with receptor-free hosts and that the bacteriocins are inactive from the outside. We evaluated the effect of the suicide probes in membrane potential using a fluorophore that is quenched in polarized membranes, and when they are depolarized, the fluorophore is released in the medium, increasing its fluorescence intensity. Here, we represent intensity over time for the different expressing strains. When we add the fluorophore, the intensity immediately increases, and when Entrecin is attached to the bilayer through ETPM, the intensity remains high because the membrane is depolarized, while both controls, ETPM and the bacteriocin from the periplasm, are not able to depolarize the membrane like the suicide probe does. With microzin B, we get the same pattern in the absence of SEAC. This suggests that bacteriocins are forming a functional pore in the absence of the receptor, but they need to be attached to do so. We use Lordan to assess the effects of the suicide probes in membrane fluidity. In the gel phase, Lordan's spectrum is blue shifted, but when the membrane is more fluid, the spectrum is red shifted. From the spectra, we get the GP coefficient. The higher the GP, the more rigid the membrane. The lower the GP, the more fluid the membrane. When Entrecin is attached through ETPM, there is a blue shift of the spectrum with respect to the controls. This is all in the absence of the receptor, and it is also reflected in the GP values. The suicide probe highly increases the GP because it's making the membranes more rigid. And this is telling us that there might be a bacteriocin-lipid interaction that is beyond the presence of the receptor. In the absence of SEAC, both microzin B attached to the membrane and free from the periplasm decrease membrane fluidity. So microzin B seems to have a tropism for E. coli membrane that, while it's not enough to produce a depolarization, it does change membrane fluidity. This is probably related with the fact that microzin B is naturally active against E. coli. To sum up, bacteriocins can depolarize the membrane even without the receptor as long as they are somehow attached. Suicide probes decrease membrane fluidity, so there might be a receptor-independent interaction between the peptides and the lipids. All this is telling us that the receptor is more likely to act as a docking molecule, and that suicide probes are actually a very interesting resource to assess bacteriocins' mechanism of action in vivo, complementing very well in vitro studies in model membranes.